what you can. So when we finished last time, we did a lot. We did a lot last time. We went through glycolysis, alternatives to glycolysis, Krebs cycle, electron transport chain, and we have a little bit to finish up on metabolism with anaerobic respiration and fermentation. There is a lot to unpack, especially with electron transport chain. I would encourage you, it's just a page or two in your book, go in and read about that again in your textbook. Here is, I guess, got a couple questions just to kind of see where you're at, see if you know the answer here. All right, this pathway, and this is, I've had a few of you question me, well, I just don't understand like what depth I need to know this metabolism stuff. I try to think big picture. I try to make you think about, well, what's special about this pathway versus that pathway? When does a cell use this pathway? When does it use the other one? What's special about what we get out of it or what we start with maybe? Okay, so here's a question from an old exam. This pathway generates electrons that drive biosynthetic reactions. Okay, so we talked about how NAD and its reduced state NADH, those electrons go to electron transport chain and get converted into ATP. Electrons that go to power biosynthetic reactions, that's going to be pentose phosphate pathway because we've got the carrier NADPH. That phosphate tags those electrons to go towards biosynthetic reactions. And you should recognize Embden-Meyerhof. That's just another name for glycolysis. We haven't talked about fermentation yet, so hopefully you didn't think that was the answer. Okay, one other question here before we get into some new content. Which one of these is true concerning the process known as the electron transport chain? Let me just give you 30 seconds to look at that and try to decide what you think the best answer is. All right, so let's go through and eliminate which ones are not true. So the first one here, it says ATP is generated due to the flow of protons against the concentration gradient. Remember, here's the cell membrane. Those protons get pumped outside the cell membrane, and when they flow down the concentration gradient from high concentration to low concentration, that's what powers the production of ATP at that enzyme ATP synthase. Okay, so that one's not correct. Electron carriers pump electrons outside the cell. Remember, those electrons have to be chaperoned, right? They have to be carried somewhere so they don't cause damage. So you're never going to be just pumping electrons by themselves anywhere. So this would be true if it said protons. Okay, and part D here, bacteria always use oxygen as the final electron acceptor. We're going to talk more about anaerobic respiration today, but they are much more flexible than that. Okay, so the answer B here is correct. FAD, H2 is oxidized to FAD. In other words, remember, this is carrying 2 electrons with those 2 protons, and both are dropped off to those carriers within the electron transport chain. Okay. All right, questions? Okay. So one other thing I wanted to revisit. We had a question last time that I did not answer as well as what I would have liked to. So we had the question about enzymes. Remember, we started last lecture. We're talking about a little bit general information about enzymes and how they can be inhibited. We talked about 2 different kinds of inhibition, competitive inhibition and allosteric inhibition. Remember the difference there. A competitive inhibitor is going to bind right to the active site of the enzyme. An allosteric inhibitor is going to bind maybe to some other site in the enzyme that then changes the shape of the active site. So we talked about feedback inhibition, where the end product of some pathway will go back and inhibit the very first enzyme, because if a cell has a whole bunch of this end product, it maybe doesn't want to make any more. And the question was, does that always have to be an allosteric inhibitor? And I said, well, I don't see why not. And I didn't, that's not incorrect, but it's almost always going to be an allosteric. I asked a biochemist, and he couldn't think of any pathways where it would be a competitive inhibitor, because a competitive inhibitor would be the same shape here as this active site. So the chances that some end product is going to be the exact shape and fit right back into that first enzyme, it's not likely, and I'm not sure if that ever would actually happen. So that's the reason that this is usually some kind of, allosteric inhibitor. So hopefully that's a little better explanation there. All right, so moving on, remember last time, I always kind of revisit this kind of big picture graphic here. We were talking about anaerobic respiration. So remember, that's the process where a cell goes through glycolysis, kind of a modified Krebs cycle, has an electron transport chain, but then there's some other final electron acceptor here than oxygen. Okay, so we were talking about those final electron acceptors and what they could possibly be. Usually, I can't remember if I wrote this, it's going to be some kind of inorganic ion. So remember, if something's inorganic, it means there's no carbon-hydrogen bonds. Like carbon dioxide is considered inorganic. Even though it has carbon, it doesn't have carbon-hydrogen bonds. Okay, so there are some exceptions there, but usually anaerobic respiration is accomplished through some kind of inorganic ion. So the example that we started with last time, nitrate is an electron acceptor. And if you've had lab already this week, you actually, this is one time where lab and lecture match up. You tested for the enzyme nitrate reductase that enables an organism to use nitrate as a final electron acceptor. So that's the so what of why we're testing for that enzyme. It allows an organism to respire anaerobically. Some other, and I just wrote out more of the equation there to try to, this is a reduction, it's losing an oxygen, so it's a reduction there, but I drew in those protons and electrons. We get a little water that ends up there too. Okay, other ions that can be used, sulfates. Okay, and sometimes, whoops, this is supposed to be minus. Okay, organisms that can use sulfates as a final end product, they are going to produce hydrogen sulfide as an end product. Okay, and that hydrogen sulfide, you may have been somewhere where you've smelled that. It's that characteristic rotten egg smell. If you've ever been to Yellowstone, smelled the geysers or whatever, that's hydrogen sulfide. So that's one of the gases that we will test for later on in the lab. Iron can be used as a final electron acceptor. Also carbonate. Oops, there we go again, 2 minus. All right, so organisms that use carbonate as a final electron acceptor actually end up with methane. Okay, sure. Okay, so the organisms that do this are called methanogens. They're Archaea. And of course, methane is a pretty potent greenhouse gas. You're going to find methanogens like in the rumen of cow's stomach. So they, you know, produce a lot of methane. Where else would you find these organisms? Anywhere where organisms are living anaerobically, bogs, swamps, also landfills. So here is, I didn't post this picture, but they can actually, if you've ever been to a landfill on a field trip, I went with my kids once, wasn't like the most interesting exotic field trip I've ever been on, but I went to the landfill just south of Grand Rapids. And it's actually pretty scientific, landfills are. They have to vent. Of course, they're, you know, covering up the garbage, you know, piling it up on top of each other. So they have to have vents because there's anaerobic bacteria that are respiring anaerobically in there, producing gases that can blow up if you don't properly vent it. And some landfills can actually catch some of that gas, a lot of it is methane, and actually produce electricity from that. And they do that on a really small scale at that landfill south of Grand Rapids. It's just not really efficient enough to be economically feasible, or you're not saving any money really by doing that. But it's an interesting idea of, again, using bacteria to do work for us. Okay. All right, so one other comment I want to make about anaerobic respiration. The book doesn't really talk much about anaerobic respiration. This looks complicated. The main point that I'm trying to get here, and I'll just tell you right off and then I'll explain you why. Of course, it's an advantage for organisms to be able to use something else other than oxygen to respire, but you are always going to get less ATP from anaerobic respiration compared to aerobic respiration. Okay, and that's kind of the take-home thing from this slide. You can certainly memorize that, you know, for next couple weeks and remember it for the exam. But to understand, I'm going to try to explain to you why that's the case, so that maybe it will stick with you a little bit more. You remember it for the final, or if you need it again for another class. Okay, so the reason that organisms that respire anaerobically always get less energy out of that process than if oxygen's using the final electron acceptor has to do with this redox tower. So basically, we can put electrodes into cells and measure the reduction potential. It's amazing to me that they can measure all this. But basically, this it's pronounced E naught prime. Okay, that's a measure of how badly substances want to give up electrons. Okay, so a measure of how badly whatever that molecule is wants to give up electrons. So for example, up here at the top, it's negative, it's very negative. So these pairs up here basically easily give up electrons. So for instance here, NADH, okay, we know that that really easily gives up its electrons right at the beginning of the electron transport chain. These pairs down here, so here we see oxygen, they have a more net positive E naught prime, so they very easily gain or steal electrons. Okay, they want to take them from other molecules rather than give those electrons up. Okay, so in aerobic respiration, you have NADH as the electron giver and oxygen as electron acceptor. So that distance from NADH to oxygen, the longer that distance is, that produces the most energy. So the greatest change in the reduction potentials gives us the largest energy gain. And you can calculate that. I would definitely leave that to your biochemistry professors. But if you look at some of the other pairs here, so sulfate, we talked about that, can be used as a final electron acceptor. That's not traveling too far, so you're not getting a lot of ATP gained from that. This is actually also how we know the order of the electron carriers in the electron transport chain. Here's nitrate, that's another, we talked about that as a final electron acceptor, so that's giving us some energy. This is an even further reduction of nitrate, so a little further. Iron, even better, but none of them are, again, as far as if oxygen is used. So again, getting a good amount of energy, more than what you'd get from fermentation, which we'll talk about next, but not as much as aerobic respiration. Right. So to finish up, thinking about what kind of organisms do anaerobic respiration. You and I do not, right? We don't have the enzymes in order to use inorganic ions. But bacteria that are considered facultative anaerobes, remember, they can respire aerobically and anaerobically, or some kind of fermentation, aerotolerant anaerobes. They typically don't respire aerobically at all. So those organisms are either doing anaerobic respiration or fermentation, and anything that's a strict anaerobe is probably doing anaerobic respiration. Okay, just not strict aerobes like us. So what's the difference between anaerobic respiration and fermentation? Those are terms that are typically confused because both these processes don't require oxygen, so sometimes they're just sort of categorized together, but they're very different processes. Okay, the difference is what happens to pyruvate. That's a key branch point at the end of glycolysis there. So basically, if there's a suitable electron acceptor, then it's going to be doing some kind of respiration. If there is no suitable electron acceptor, sorry, just wave your hands at me if you can't read my handwriting. It gets a little sloppy trying to write like this. If there's no suitable electron acceptor, then the cell has no choice but to do some kind of fermentation pathway. Okay, so let's talk a little bit about fermentation now and what that will involve. So I came back to this graphic and I wrote out kind of a dictionary definition of fermentation. Okay, so it's the orange parts of our diagram here that's a fermentation pathway. Okay, a redox process in the absence of oxygen in which the substrate is partially oxidized and the final electron acceptor is an organic molecule. So let me just underline that. Final electron acceptor, almost always something organic, almost always something already in the cell. So a very common electron acceptor for fermentation pathways is that pyruvic acid. Okay, it's right there in the cell already. The pyruvic acid, let me just write that. Pyruvic acid or pyruvate accepts the electrons from the NADH. Okay, that's coming off here of glycolysis, and then the NADs can cycle back to glycolysis and at least keep glycolysis going. If there's nowhere for these electrons to go because there's no electron acceptor down here, then even glycolysis will come to a stop. Okay, with the redox reaction, you have to have, if you don't have the oxidation back to NAD, then you can't have balancing reduction. So you have to have that NADH cycle back to NAD just to keep glycolysis going. Okay, so oftentimes it's the pyruvic acid that accepts those. And so here's an example of a couple of very common fermentation pathways. The very simplest fermentation pathway is what's called a homolactic fermentation, where pyruvic acid accepts those electrons here from glycolysis and just is converted into lactic acid. And that's all. This diagram is a little bit confusing because it makes it look like, oh, the cell could make lactic acid or the cell can make ethanol. Typically, a cell will do one kind of fermentation pathway and not have a choice of lots of different pathways that it can use, because we actually use that as to what fermentation it does to help us identify organisms. So this is a little confusing. It's not going to go one way or the other. This is an example of one fermentation pathway that organisms like Streptococcus use. It's the simplest one, and it's one of the only ones that there's no gas produced. Interesting enough, no CO2 produced. Okay, and that's the pathway that we can do to some extent in our muscle cells. All right, so if you've ever run a 400, you know, you start coming around that final turn, you got lactic acid building up in your muscles. It's getting harder and harder to move. Okay, so we can do that to some extent. There's some bacteria that's all they do. And what do you notice about this pathway in terms of how much ATP you get? None, right? So the only ATP that organism gets is a little bit of ATP that they get out of glycolysis. So they have to burn through a ton of sugar to get enough ATP just to run their cell. There are some fermentation pathways where a little bit of ATP, I'll just write that here, is produced during some fermentations, but it's only a little tiny bit compared, especially when you think about how much ATP, like 36, 38 that we gained from aerobic respiration. Okay. This other fermentation pathway, and where you end up with ethanol, it's mostly yeast that are doing this. There may be some bacteria that do that, but this is mostly yeast and alcoholic fermentation. You can see very common to have a little carbon dioxide or some other gas as a byproduct there. And just because I like for you to have something interesting to discuss with your roommates or your parents, check out this little article here. It's not particularly scholarly, but it's a little article about a syndrome that's been described in a small number of people, a handful of people, and they call it gut fermentation syndrome. So they have found people that have an overgrowth of yeast in their large intestine. We all maybe have some there, but maybe some people have a whole lot more. And after they've eaten a diet high in sugar, you know, whatever, they ate a lot of sugar, and yeast were fermenting that sugar into ethanol. They've tested people, somebody was 4 times the legal limit of blood alcohol, and he hadn't had any alcohol to drink. And they found that he, again, had this overgrowth of yeast. So, you know, I don't know if that would work very well if you got pulled over by an officer and said, I haven't drunk anything, it's just, I got yeast. I don't know if that would work or not, but it might be worth a try. Okay. All right, I did not post this because I didn't want to scare you. All right. To me, fermentation is incredibly interesting. I don't, you know enough about the pathways to be able to write them out or whatever, but I just wanted to tell you why this is so interesting to me. We get lots of useful products from bacteria because they do fermentations. So again, it starts with glucose, typically some other kind of sugar, and then pyruvate, the end of glycolysis. And then again, there's a whole different variety of different fermentation pathways that organisms can do. We talked about this lactic acid fermentation. Here's one of my favorites over here that the Prokaryotic Bacterium do. They end up with an end product called propionate. That is the distinctive taste of Swiss cheese. And the carbon dioxide that's given off, that's what makes the holes in Swiss cheese. In lab, you'll be testing to identify certain kinds of bacteria, whether they do this 2,3-butanediol pathway, or whether they do a mixed acid fermentation where acetic acid is one of the end products. So we'll talk a little bit about that in lab a little bit later. Some of my favorite pathways here, Clostridia Bacterium. So we talked about Clostridium briefly. That's one of the genera of organisms that can produce spores along with Bacillus. And the Clostridia are unique because a lot of them make really nasty neurotoxins, so really nasty diseases. One of them is C. diff, Clostridium difficile. And this intermediate in their fermentation pathway, butyrate, is what's responsible for the distinctive odor of the diarrhea of somebody with C. diff. And apparently there are some nurses that say that they can diagnose a patient whether they have C. diff or not just by smelling their diarrhea. It's called the whiff of the diff. All right, so you people that want to be nurses, this is what you're getting into, just preparing you. It's this butyrate. This is also the very distinctive odor of vomit. All right, so nasty stuff there. Somebody told me once, I really just am not interested in microbiology, and like, how can you not be interested in this? It's, you know, smelly stuff, but it's kind of cool. The whole deodorant industry is based on bacterial fermentation, so some kind of nasty smelling organisms. So, okay, and next week in lab, if you're in lab, just trying to tie in lab for those of you that are in it, you're going to be inoculating some fermentation tubes. So you're going to have different tubes that have different sugars in them, like glucose or lactose, mannitol, and see if the organism that you inoculated in there is able to ferment that particular sugar. Now, we're not really keeping the oxygen out here, but that's what organisms, they might be respiring a little bit too aerobically, but they're going to be running through that sugar and doing fermentation as well. And you always get acidic end products from bacterial fermentation. So you get a yellow color change if there's acidic end products. So we're just looking to see if the organism, you know, can use certain sugars or not. We also test for gas production. There's a little tube in there, it's hard to see from this picture, called a Durham tube. And the gas bubble, again, is sort of hard to see there. There's no gas bubble here. Just another way to characterize, help us know, oh, it's using a fermentation pathway where you get gas, or here this organism is maybe fermenting a little bit and not getting gas production. So anyway, you'll be inoculating that last week. And lastly, I just want to make the point that, you know, we talk a lot about how bacteria make us sick, and we focus a lot on that, but they do a lot of good things for us too. So I don't want you to memorize what organisms do what, or memorize these products. Just appreciate the fact that bacteria do some good things for us, right? We wouldn't have Swiss cheese without bacteria. We get, you know, other kinds of cheeses, yogurt, soy sauce from different fermentations. Of course, alcoholic beverages, that's yeast that do that. Things like rubbing alcohol or nail polish remover that you wouldn't think could be a product of a bacterial fermentation. Of course, vinegar. So all sorts of useful products from bacteria there. And this is, I just mentioned, this is just a nice little table kind of at the end there of chapter 5. Some of you have, you know, told me that you studied a lot for the first exam and it didn't pay off. Like, you didn't do as well as you wanted to. As much as possible, you need to be kind of quizzing yourself as you go along and trying to see, do I understand this? So this is a great table that you would want to maybe cover up part of this. Let's see, aerobic respiration, is oxygen required? Yes. Okay, so cover that up. It's good, just kind of general big picture stuff. Quiz yourself and see if you know the answers there. So good review there. All right, questions as we move on. All right, so it would be great if you could start working on that chapter 5 homework if you haven't done so already. All right, so our next big unit that we're going to spend probably about 3 lectures on, and I think we're pretty close to being on schedule, is microbial genetics. So we did leave out parts of the book from chapter 5. We didn't talk about photosynthesis. We didn't really talk about anabolic reactions very much, but chapter 7, we're pretty much doing the whole chapter. I like to bring in some other graphics to kind of help explain a few things a little bit better. I don't think the book does a great job with here. But we're going to be thinking about the structure of DNA. We're going to start with that. A little bit about the form that takes in prokaryotic cells. We should get to replication today. That's how DNA is copied. Probably next time into how RNA is made, transcription, and how proteins are made from messenger RNA. That's translation. A little bit about regulation. It's very expensive energetically to make proteins. So how does a cell keep from making proteins that it doesn't really need? And then we will finish up with a little bit on mutation and just describe some different kinds of mutations and things that act as mutagens. All right, so I like to start out with, in the past, I've had students sit and just see if they could sketch out the structure of DNA. And I decided to just change that into a little multiple choice question. Don't look at your next slide. So without looking at the structure of DNA, see if you can answer this question accurately. Which one of these accurately describes the structure of DNA? So I'll let you read that for a second. All right, so hopefully, if you had to draw the structure of DNA, hopefully you could draw a nucleotide. Okay, here's the sugar, and here's the base, and here's the phosphates. Okay, so the only one of these that could possibly be true is number B, or letter B, let's call it. Whereas sugar is bonded to a base and a phosphate. Okay, you never have the phosphate bonded to a base. You never have 2 bases bonded together on the same strand. Okay, they bind to their complementary base on the other strand. You would never have a base bonded to the phosphates. Okay, and of course, a nucleotide, we would call it a ribonucleotide if it was RNA, and you would have ribose sugar. So, all right. So that, I don't know if that was hard or easy for you, but let's look at the structure of DNA. This should be review. So let's see. Of course, we're talking about the genome of a cell. That just means all the DNA in a cell. And if you look at the structure here, like I said, we've got these 5-carbon sugars, deoxyribose sugars, forming what we call the backbone that alternates with the phosphates here. And the 4 bases that we find, nitrogenous bases that we find in DNA, adenine and guanine are purine bases, and the pyrimidines are thymine and cytosine. Okay, so nitrogen-rich bases. Right. And if we look at the structure a little bit deeper, again, this is right graphic from the beginning of chapter 7 here. Obviously, I don't need you to memorize the structure of those bases or anything. Double helix structure, Watson and Crick elucidated that in 1953, wrote a one-page paper describing it. They had a lot of help. Other people contributed to that, but they got the credit for it. Notice the directionality of that double helix. We call it anti-parallel, because we have one end is considered a 5' end, and that is going to match up with kind of the opposite way on the other strand, the 3' end. So those names, 5' and 3', remember that refers to the carbon. So if you look at the numbered carbons here in the deoxyribose sugar, you start counting the one carbon is the one that's attached to the base. And then you start counting away from that oxygen in that ringed sugar there to 3. So the 3' carbon, this is going to be the 3' end of the strand. For 5, that carbon is going to be the 5' end. Okay, so then the other strand, here's the number 1 carbon attached to the base, and then again you're going away, numbering away from that oxygen to 3. So this is the 3' end, and for 5, this is going to be the 5' end. All right, so that directionality gets a little bit confusing, but we will talk more about that. A nucleoside is the base and the sugar. So deoxyribose plus a base. And then a nucleotide is the base and the sugar and the phosphate. That terminology can get a little bit oppressive, but if you use that correctly, nucleoside, nucleotide, that's the difference there. So how is the DNA, what form does the DNA take in prokaryotic organisms? A little different than eukaryotes. So in prokaryotes, we typically only have one chromosome. I think maybe we mentioned this before, and we were talking about cell structure. Usually one chromosome, and it's usually circular. Okay, and if you contrast that with eukaryotic DNA, they typically are going to have many chromosomes. Of course, we have 46, we have 23 pairs, and it's usually going to be linear. So the advantage to the circular chromosome, rather than the linear one, circular chromosomes are going to be a little bit more stable because they don't have their ends exposed. The ends of the chromosomes are going to be subject to nucleases that are present in the cell. So if you're going to have a linear chromosome, you have to have a way to protect the ends of the chromosome so they don't get degraded. So it's just a little simpler to have a circular one. And there are exceptions there. There are bacteria with 2 chromosomes or maybe more than that. Some have linear chromosomes that we found. The prokaryotic chromosomes are always going to be haploid. Okay, so that means 1 copy of each gene. And of course, eukaryotes are always diploid. We have 23 pairs of chromosomes. Okay. And where's that DNA found in prokaryotic organisms? Found in a region of the cell that we call the nucleoid region. All right, so that's down here. Remember, it's not enclosed in a membrane. And of course, for eukaryotes, those chromosomes are going to be found in the nucleus. But remember, the DNA of mitochondria and chloroplasts are found, are in that prokaryotic form. Okay, so they're circular. All right, so in addition to chromosomes, bacteria, as well as archaea, a lot of similarities with bacteria and archaea. Archaea actually store their DNA a little bit differently. They use histone proteins, more like eukaryotes. But bacteria also have plasmids in addition to their chromosomes. And plasmids are just small molecules of DNA. And they are circular also. Okay. They replicate on their own timetable. They replicate independently. Right. They replicate independently of the chromosomes. Okay, and they're going to carry genes to replicate themselves and for some other task that's not absolutely necessary for the survival of the cell. All right, so carry genes to replicate. And some traits not essential for survival. So in other words, you would not have the enzymes of glycolysis encoded for by a plasmid. Okay, that glycolysis is absolutely crucial to the survival of the cell. What kind of things might you find on plasmids, or what are some examples of different kinds of plasmids? Let me change colors here. An F plasmid, that stands for fertility plasmid, is going to carry instructions for conjugation. And we haven't talked specifically about conjugation. We will get to that. It's a kind of horizontal gene transfer. But we have talked about organisms that can do conjugation, have a particular structure called the pilus. Okay, so we'll say they are able to make or form a conjugation pilus. And we have talked about that. Our plasmids, that stands for resistance. Okay, carry genes that help them withstand or live in the presence of some kind of antibiotic. Okay, we'll be talking more about that a little bit later as well. There are plasmids you probably haven't heard of, bacteriocin plasmids. Organisms that have bacteriocin plasmids can secrete bacteriocins. These are substances that kill cells that are the same species or a very similar species to that cell. So bacteria are sort of similar to humans in that respect. Sometimes they work together, like in biofilms. Sometimes they kill each other. Okay. Some scientists don't really agree on whether to call bacteriocins a very narrow spectrum or very specific antibiotic. But sometimes they are considered that. Okay. And then virulence plasmids. We will be talking soon in another few weeks about virulence factors. These are just anything that a cell can do in order to make it more successful to form an infection or to cause damage in a host. So a virulence plasmid would carry some gene to make the cell more successful in establishing some kind of infection. And oftentimes that can be maybe a toxin. It can maybe something that makes it withstand our immune response a little bit better. Okay, so there's just some examples of different kinds of plasmids. If the cell couldn't make those, it wouldn't necessarily die, but it's just gonna make it, you know, maybe more suited to live in certain environments. All right, I'd like to spend a little bit time here. Watson and Crick, when they wrote their famous paper elucidating the structure of DNA, they didn't do any experiments or really make any comment as to how they thought DNA is replicated. So at the time, there was, you know, this is mid-50s, there was 3 hypotheses as to how DNA might be replicated. And it took a very famous experiment to try to figure that out. So the 3 hypotheses at the time were that replication is semi-conservative. And that means in the daughter, you could say, DNA, one strand of the old was conserved, and then there was a new strand. Okay, in black and white, the next few slides are going to be a little bit harder to figure out what's going on. A conservative replication means that the original strand here was conserved, and then one of the daughter strands was totally new, was made from that old strand. And then there is a dispersive hypothesis that maybe every, you know, so often in the DNA, it would be chopped up and you get, you know, a mix of new and old, and just kind of pieces. All right, and the way we figured out which of those methods was correct was by a very famous experiment I want to talk a little bit about. It's called the Messelson-Stahl experiment in 1958. Yep.
What did you say about the conservative strand?
So the reason that it's conservative is that basically the whole original strand was conserved, and then the copy of it is all new. So like basically this strand would unwind, and then you would somehow get a whole new strand. Okay. Stay tuned for which is correct. Right, then it will make more sense. So a very famous experiment, so I want to go through it a little bit more carefully. This has been called the most beautiful experiment in biology, because not only did it prove which of those 3 methods must be right, but it also disproved the other 2 methods and why they can't be right. All right, so Matthew Messelson, Franklin Stahl, 1958. And I do have, I'm gonna walk you through how this works, but I do have a link to a YouTube, well, it's from a textbook. It's like a McGraw-Hill animation. It's just a couple minutes long, and it walks you through, probably, you know, explains it even better than I will. So there's a video link here to explain it if whatever, something that I say doesn't make sense. But the gist of this experiment is that they grew, I don't know what organism it was, maybe it was E. coli, I'm not really sure, but they grew some bacteria in some heavy nitrogen, some media with heavy nitrogen. It wasn't radioactive. I think it's nitrogen that has an extra neutron. Normal nitrogen is N14, so media with N15 in it. Okay, so they grew it for a few generations that way, and then they switched the cells into a medium with regular nitrogen. And they timed it and tried to time it just for about 1 generation, and then they took the cells out and they centrifuged them. So if they centrifuged the cells that were grown on the heavy nitrogen, you would get a band that's pretty low in the tube here, a distinctive spot. If after the first generation, so remember these are cells that were switched to light nitrogen, you would get a band that's a little bit higher, a little bit lighter than the original one. And then if you took some cells out after 2 generations, now you're getting, still some hybrid DNA here, so you get this hybrid band still, but then you've got some cells that have only been grown on that lighter regular nitrogen, so you get a band that's even lighter. Okay, and then if you kept going, this hybrid band would get even thinner and thinner and thinner, and you get more and more of DNA that's just been from the light nitrogen. All right, so basically, what does that mean? How can we interpret that? Well, if the conservative hypothesis was correct, what would you see in that experiment? You would see here that first generation progeny, you would have a heavy band and you would have a light band. You would have 2 bands, which is not what we saw. We saw a hybrid band right here from the first generation. So right after that first generation that this conservative method can't be correct. Here we've got, again, hybrid band, hybrid. If dispersive was correct, you would also have 2 hybrid bands. So you have to go out another generation to see which it is. If you went out another generation here with the semi-conservative, you would still have a hybrid band, but then you would have, again, these would be a light band, higher in the column. But if dispersive was correct, you'd still be having a bunch of hybrid bands and only hybrid. Okay, so again, and that's not what we see. We see the light bands and less of the hybrid. So again, the experiment proved that semi-conservative must be correct and the other 2 not. Okay. So again, that's website there. Check that out. All right, what's going on at more of the molecular level? That's sort of, big picture. Okay, it replicates semi-conservatively. What's going on at the molecular level? So DNA is a polymer, right? It's made up of many different monomers, right? So those monomers have to be pieced together. And again, this terminology gets a little bit repressive, but we talked about a nucleoside is the base and the sugar. Nucleotide is the base, sugar, and phosphate. Well, actually, the monomers that get added together to make the polymer DNA are these triphosphate deoxyribonucleotides. So basically, you have the base and the sugar and 3 phosphates. Those phosphates get cleaved off, and that makes it a very energetically fast reaction, adding those nucleotides together. Okay, so again, the terminology is cumbersome to me, but when you see this DATP or DGTP, that stands for guanosine triphosphate deoxyribonucleotide. All right, so the deoxyribonucleotide is the D part, and then guanosine triphosphate. Okay, so we would have adenosine triphosphate deoxyribonucleotide, or cytosine, it's cytidine, I think is the way they say it, triphosphate nucleotide, or thymidine triphosphate deoxyribonucleotide. Okay, so they carry with them those monomers the energy in order to make the polymerization reaction. So if you have, of course, here's an existing DNA strand. Of course, it's going to be You know, you're going to have a template strand here with T, G, A bases here. The new triphosphate nucleotides are always going to need a free hydroxyl group here, a 3' hydroxyl group, in order to add the next triphosphate nucleotide on. Okay, so there's an enzyme that catalyzes that. We'll talk about in a minute, DNA polymerase 3. 2 of those phosphates are cleaved off. Again, it makes it very energetically favorable. And we get another one added on and make the DNA strand a little bit longer here. So if this was a G, this would be a C, and then that's kind of matched there. Okay, so it's easy to forget that, oh, there's actually 3 phosphates on there. Kind of forget that. Oh, some of my slide got cut off there, sorry. All right, so where does DNA replication begin? Remember, we've got, let's say we've got our chromosome, we've got our circular structure of DNA that needs to get replicated. Okay, replication begins at a very specific spot on that chromosome. And it's a spot called the origin. Okay. Sorry, that's an I there, origin. Okay, so this little green box here, that is supposed to be the origin. How do the enzymes know that that's where they need to start replication? There's probably some signaling that happens there. Perhaps some methyl groups added. We'll talk about that a little bit later. But it also might be a section of the DNA that has lots of A's and T's, because I kind of skipped over this before. There's 2 hydrogen bonds between those bases, the A's and the T's, and there's 3 hydrogen bonds between C's and G's. So if you had an area with lots of A's and T's, the enzyme that comes in and basically breaks those bonds can do that a little bit easier, okay? So that enzyme is called helicase. Usually we refer to that as unzipping the 2 strands. And you'll see that on the next page, the helicase. So I'll just, okay. Okay, what else? Prokaryotes usually have 1 origin. Okay, like you see in the picture here. In eukaryotes, because they have so much more